We're going to initially assume that you know nothing about viruses or cell biology, but we're going to explain those things rather rapidly and then get pretty deep into the technicalities of both of those. So let's just cut right to we're going to interweave biology, the molecular biology with the virology, because it's hard to understand a virus, which is a process, without understanding the cells that are actually making this happen or facilitating it. And so this is everybody's favorite conundrum in biology, is what is a virus, because it's actually not so easy to put your finger on. I defined it here based on a number of different definitions. This is something that I think works, which is I've said that a virus is a process that involves the production of infectious nucleic acid containing particles by living cells. So viruses are always made by somebody else. The virions, the little physical objects, are always constructed by cells that are out there, which makes them very strange infectious agents, because normally we think infectious agents as these little microbes who are out there trying to look out for themselves. And it's not so clear whether viruses are trying to do anything whatsoever actively, because although they can be selected for in a traditional evolutionary fashion, they don't do anything actively. They're not mobile. They don't reproduce actively. They're essentially a process that's happening. They're like a piece of software that's going on inside of living systems. So we haven't known about viruses for a very long time, which is also really interesting.
Part of that is because they're so small. They're significantly smaller than bacteria. They're significantly smaller than fungi, than worms. If you're doing a normal preparation and you try to filter some kind of supernatant, you can easily filter bacteria out with the sorts of membranes that were available 1900 and earlier. But when you're trying to actually deal with the presence of a virus, they slip through the pores of most membranes very, very easily, to the degree that even today, most labs aren't working with isolated virus. Most labs are working with cells that produce the virus, and so you put a vector into the cells, and then the cells start to produce it, and then you have gobs and gobs and gobs of viruses. But there's very, very little research that's being done where someone's like, well, we took this one virus and we looked at it. Because these are basically at the resolution of the wavelength of light. You usually are looking at these using...
Well, they're much smaller, actually, than the wavelength of light, which is crazy.
So what are people imaging them with?
Well, people aren't usually imaging them, which is what's crazy.
Well,
I mean, they do, but if they do, they're coding them. They're doing it with electron microscopy. So, which is actually like a field-based way of, like, to do electron microscopy, you coat something in metal, and then you sort of see how your electrical circuit interacts with that metal. But to give you some idea for the size of these things, viruses can go as small as like 20 nanometers, right? And 20 nanometers is, let's say, a bacteria is about 1,000 nanometers, perhaps. So you're talking like 50 times smaller than a bacteria. Bacterias are probably 20 times smaller than a cell. So these are just they're unbelievably tiny organisms, or they're not organisms. They're unbelievably tiny particles. And the original research that was done essentially demonstrated, especially going back to this one that's written on here in 1898, which was the tobacco mosaic virus. They basically were just finding that there was some substance being produced by the organism that was infectiously transmissible, and it wasn't a microbe. They couldn't see it. And so for many, many years, nobody really knew what was doing this. But they referred to these processes as viruses, despite the fact that they couldn't see anything. And they didn't image it for many decades until they had the adequate technology. So it's actually a really, now that I think about it, that's actually a really cool scientific triumph, which is the prediction of these physical objects before you're actually able to observe them. I'm sure there are, that's a very rare and beautiful thing to have happened. You see a lot of folks doing that in physics today, making predictions of objects that they can't see yet. And so this is sort of a testament to the fact that it is possible. At any rate, the first coronavirus was not discovered until the 30s. And again, they weren't taking pictures of them. This was merely a fact that these little chicks were getting sick, and they could take the effluvium from the chicks. Like something.
By the way, when he says chicks, he means literal chickens.
Yeah, like little chickens, chick babies. And they could transmit this disease, but they couldn't detect any microbes that were causative, that were associated with it. And so they were like, well, it's a virus. And that first coronavirus was killing all these baby chickens, but the adults were not affected by it. It was kind of interesting. Anyways, the viruses can mutate just like organisms, and they can be selected for or against through those mutations, which really confuses people because it makes them be like, are they alive or what's going on? But really, any non-living system can evolve too. You can just set up completely algorithmic evolutionary systems too, where you just enforce some rules and decide whether this variable gets kicked in or out of the system based on what you're selecting for. So that's viruses in a nutshell. A really wild process. I think it's worth also spending just a second to point out that viruses get a really bad rap in the world. Let's see if I have a slide for this. Yeah. So viruses get a really bad rap. We normally think of them as terribly infectious, horrible processes that are wreaking havoc on our lives. But I think that this underestimates the utility of viruses in biology at large. So this is a New York Times article, which I thought was a really funny headline. But it's true. There's gazillions of viruses coding everything all the time, everywhere. It is hard to overemphasize the presence of viruses in our daily life. Viral particles are everywhere. And so this has a lot of people scratching their heads like, well, if these viral particles are everywhere and people aren't dropping dead everywhere, what are they there for? Why are the cells making them? Are they doing anything? And without getting completely sidetracked into it, some of the more interesting utilities that have been proposed concern horizontal gene transfer. Do you want to say anything about horizontal gene transfer? The real question is, if everybody is, how do you, in order for people to, or any organism to become more well adapted to its environment, it has to change over time. And how do you get changes if reproduction is just perfectly
fidelitous?
Yeah, if there's no mistakes, and if everything's just the same from generation to generation, and you look exactly like your parents and they look exactly like their parents, how do you actually get adaptation, right? Because you're not really experimenting with the next progeny at that point. And so some folks are proposing that the role of viruses centrally in biology is not to just kill stuff. The role of viruses is to generate diversity by shuffling genes, essentially. And this is supported by several lines of evidence, but I think the most convincing is the presence of viral genes in mass in the genome of organisms, including ourselves. So for many, many years, people thought, oh, well, there's like all this, the genes only represent a small portion of the DNA. The rest of this DNA is just junk. And they slowly began realizing that, oh, these are actually incorporated retroviruses. These are all ancient viruses that have been put back, or they're DNA viruses that have been shuffled back into the genome of the host. And so, I I think it's really worth considering as we go through all of this that while viruses are sometimes have unfortunate consequences, especially with regards to immunology, because what we're going to find very soon is that a lot of the pathology in COVID has to do with the way your body responds to this transmissible agent. Some of it does actually appear to be a result of the transmissible agent causing problems, and viruses can produce lytic phenomena where the cells are actually destroyed. But a lot of the damage comes from the body meeting the transmissible agent and being unhappy with it being there and trying to, not have it happen.
Yeah, we'll cover that next week. We have a really good paper that's on deck about basically the molecular biology of COVID and the arc of the immune response. But before you move on about viruses, a note about horizontal gene transfer, where basically horizontal gene transfer is what creates great difficulties when you are attempting to reconstruct the lineage of organisms like bacteria. And so it's hugely widespread. It's a known problem because basically what happens is that if you have two bacteria that are compatible with one another, they basically do this thing called conjugation. And what that means is that they extend these little sex pili to one another and they pass plasmids back and forth. And what happens is that if you have an E. coli with one genome and it conjugates with a Pseudomonas aeruginosa, they won't exactly exchange chunks of the main plasmid DNA, but they will exchange small pieces of circular DNA that can be inserted into the genome of the respective bacterial species. And viruses can do something similar with humans on some level. And this is still very theoretical. This is still it's almost at the edge of evidence-based science and is more on the realm of things that we will in the future be able to see. So it's prediction. But the idea is that if you have a virus that's replicating, and if you have the viral program that's running full force, the cell is a very messy area. And so it's possible to grab genetic material, whether that is RNA or that is chunks of double-stranded DNA, as you're being packaged, because these things get sent to the wrong area, or as your virion capsule assembles, that you're basically packaging something into it that shouldn't be there. And then when you go and you move on to a different host, you carry that with you. And in most cases, this doesn't go anywhere because you get to your new host, you infect some cell that is a somatic cell. That cell lives its life. Perhaps it gets mutated by it. It dies. Perhaps it turns into cancer and the host dies, but there's a dead end there. But if you get this horizontal gene transfer to end up in the gametes, where the virus actually targets reproductive cells, then you have that it has the opportunity of getting fixed in the population. And it could be that this does nothing good. Most of these mutations will do nothing good. But it is possible for it to happen, to be passed down to the next generation, and therefore to do, as Dr. DeLay says, to increase variability of the species on that level. And that's pretty far out. Like, these are not events that are common, because as you can imagine, if you have horizontal gene transfer from this thing that's just shuffling genes, most cells are going to get real sick from it. Like,
I'm not sure. Yeah,
massive changes to the DNA usually. It's the end of days. Yeah.
That's interesting because I feel like there's so much non-coding information in this cell. That I feel like these things could happen in the background constantly, and we wouldn't even notice them happening.
That's true.
I mean, look at this New York Times headline: trillions upon trillions of viruses falling from the sky every day. But I think my imagination tends to think of the wider organismal community as recapitulating what we see at the cellular level, and the cells are communicating with one another through these different chemicals. Why shouldn't the organisms be communicating with one another through different, messaging structures? And so,
you know that this occurs because if you look at something like plants, you know that if you have a group of acacia trees and there's predation upon one of the acacia trees, it's capable of sending out a signal. I think that in this case, it's a signal that travels through the air. It's basically like an airborne hormone that induces the other acacia trees in the environment to make their leaves more bitter in order to decrease the likelihood of predation. I've heard that you can play the sound of caterpillars chewing leaves and cause them to release these stress hormones. So these are like
chemical message versions, right? And it's not a huge stretch to imagine there being some communicative activity going on. Because there's also this parallel structure that's probably worth mentioning too, where cells within your body will communicate with one another through very similar processes to the virus process.
Called exosomes. The difference generally is that exosomes don't have capsids. They're basically chunks of membrane that enclose genetic information.
So they look a lot like a virus. They're just not as well suited to exist outside of the body. I think
they're less transmissible.
The protein capsid protects that nucleic acid so that it can travel across regions of space.
Can you define space?
The distance between objects. All right, so here we are. We have viruses everywhere. They're kind of mysterious. We've only been studying them for like 100 years. So and this is something that gets buried all the time, is we really have, in the modern sense of science, been only at this for a little while. So it's totally understandable there's gaps in our knowledge. We're trying to make sense of this. We have a pandemic going on, trying to understand what's happening. There's been other pandemics when viruses were even less poorly understood.
There's also a gradual sense that our understanding of what viruses might be is shifting. Like, we're going to talk about the biochemical ways that, you have the spike protein and it interacts with this receptor and it gets into the cell and then it activates this program. Okay, great. But there is sort of a big shift that is on the horizon when it comes to understanding viruses as being pathogenic versus not pathogenic. There are something like 30 million species of bacteria, and there are, I think, between 100 and 300 pathogenic strains that are harmful to humans. And almost all of the research is done on the pathogenic ones, and almost to exclusivity was there a focus on the fact that bacteria are bad for you, which is why Lysol was really popular, hyper-sterilization of surfaces, this desire to scrub the world clean of microbes. And we're only now, after 20, 30 years of doing that, are starting to realize that, hey, we really, really screw people up when we do that.
And it gets even worse. I want to just reiterate what you said. Although there are even though there are very few pathologic bacteria, even within those pathological bacteria, pathogenic bacteria, even within those pathogenic species, not all of the patients that are infected are going to even get sick from those, and that's what's really crazy too. And so a lot of these terrible infectious agents, they'll make most people sick, but some people don't get sick, and so it's just not clear that it's downright untrue that their job is to cause disease. That is a very minor part of what microbes and viruses do. And so we have to be careful.
And this holds for even things like Ebola. Like, if you start looking through the Ebola literature, you suddenly start to realize that people are like, okay, so there's definitely a big fraction of people who they get Ebola and it's the full thing, like bleeding out of every orifice, all their endothelial cells lyse. Like, it's just the most gruesome, horrifying way to die. And then they start to do immune surveillance of the community, and they're like, there's a lot of people in the community that have antibodies for Ebola, and they had no symptoms. They had nothing happen to them. And so there's great variability in what it means to be exposed to a virus, to allow it to replicate inside of you, and then the act of your body reacting to that imposition. And so it's very sort of nebulous. There's a video here.
So before we play this video, so the purpose of playing this video for you, it's like 5 minutes long. I think it's worth watching because we need to at least all be on the same page about how cells do their job. well, cells essentially do the job of building themselves and building pathways to talk to one another. And sometimes they move themselves around, but generally they have some goals that they're trying to achieve. And the way they do that is by constructing proteins, essentially, because they use the proteins to talk. They use the proteins to, fortify themselves and everything else. And so what is called the central dogma of biology is this idea that you go from a gene to a protein in this very particular way. And so I love animations, so we have a couple of them to check out today. And I think it'll really just sink it in your brain what is happening. And once we understand what the cell is doing, then we can understand how the virus takes advantage of this process. I don't even know if that's a fair way of putting it, but how the viral process integrates with the cell's machinery in order to result in various outcomes, very few of which are pathogenic. But
here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
And that is the story of biology, at least in a simplified fashion. Let's go to that next slide here. So what you've just heard is called the central dogma of biology. And now that we understand that, I think we can start to think about the different types of viruses and how they make use of this.
Number 1 is RNA splicing. RNA splicing is a mammalian process. It is not found in bacteria. So that's just something to remember. It's a much more complicated process of going from DNA to protein in mammals than it is in bacteria, because you basically have, like she said, all of these recombinations of the RNA in order to get the thing that you want. And so a lot of proteins, the variety of proteins is produced not because you have a bunch of different genes, but because you have a different splicing organization, and so you get different proteins that end up looking that end up looking different, despite the fact that the initial RNA strand that was transcribed is exactly the same. The second thing that I want to say is that this aspect of the protein folding, she completely glosses over at the very end of it, where she's like, okay, you have the ribosome, the amino acids releases from the ribosome and it folds, and then you have a protein. The process of folding a protein is very, very complex, and as we're going to discover in this paper, it actually has a lot to do with the sort of the damage that the virus... Oh, never mind. No, sorry. Not this paper. It was a different paper. It was the CBD paper. The reason that their CBD is able to prevent serious cellular disease in mouse models in cells that are infected with SARS-CoV-2 is because it upregulates the ability of the cell to deal with protein folding stress.
So somebody asked about CBDs in the last lecture, after the last lecture, just for folks who are watching this.
Just like out of nowhere. But so basically what it looks like is that, and this doesn't hold for THC. If you include THC in the mixture, it actually decreases the efficacy of this. And none of the other cannabinoids do this. It's just CBD. What happens is that it activates gene expression inside for the endoplasmic reticulum such that it makes the stress response to overproduction of proteins much more robust, and the cell doesn't accumulate a bunch of aggregated misfolded proteins causing it to then die. Because if you don't fold your proteins properly, they stick to each other, and if they stick to each other, they make other things stick to each other, and then you basically have literally just gummed up the works of the cell. And so CBD apparently works on the basis of preventing that from happening, and it was a crazy paper. If you have time, it's pretty straightforward. It's not that long. Like, very cool mechanism. I'm done. Sorry.
Cool. So I think we're ready to get into the specifics of the SARS virus, which is our what should we say? Our
point
VIP.
Yeah. Our point of
very
order.
Important virus. One thing I was just checking out, I was just looking into Ebola really quickly, just to say one more general thing about viruses. They often are endemic within a species and don't seem to be causing a lot of problems with that species. And then once they end up in a species that they're perhaps not intended for is when you get some of these crises.
So do you think that they're trying to like make fruit bats out of humans?
I think that it might be more like maybe the Ebola virus does something productive within that population, but then once the humans acquire that, they get bat instructions, and the bat instructions are not only not productive to the human cells, but actually confuse them to the degree that they're incapable of surviving.
I expect your research proposal on the matter to be on my desk by February.
Yeah, my free time. All right, so with let's go on and let's look at SARS-CoV-2. SARS-CoV-2 is a positive sense RNA virus. So what does that mean? So there's 4 different types of these are essentially, this is the family tree of viruses, which is kind of
RNA viruses.
RNA. Well,
there's also a family tree of DNA viruses.
This is no DNA virus?
No, it's just RNA.
Oh, bummer.
I have another slide for RNA.
Okay.
Or
Wow,
DNA.
we should have put that first. But what's the point is that how do you group the relatedness of different viruses to one another? Well, just like everything that we look at genetically, it's all by homology. It's who shares what with who. And so that's how we end up with these different types over here. There's basically the basic difference between the positive sense and the minus sense is that the positive sense RNA viruses are ready to go. So their RNAs can go straight into the translation step of that dogma, whereas the negative sense ones need to use some of the host machinery, or maybe they bring it along in order to produce that positive sense RNA. Do we have a DNA? Did you say you inserted a slide for DNA?
Yes. There's a couple of other things that I want to say here where it's like as you're looking at these different constructions of viruses, so you have the positive RNA virus, like Dr. DeLay said, comes in, immediately is able to be transcribed into the proteins in question. Retroviruses have a step where they basically go into the nucleus. Negative strand RNA viruses have a pretty complex life cycle where once they get into the cell, there's a bunch of different ways in which the genomic material is processed. And then it has part of the life cycle is inside of the nucleus. Double-stranded DNA is not even...
RNA.
Sorry, double-stranded RNA is capable of also being transcribed inside of the cytoplasm, because if you have a double-stranded RNA and you unzip that, that means that you have the positive strand RNA that you need. If you only have the negative strand RNA, which is over here on the left, you end up in a much stickier situation where you have to make your way back to a positive strand RNA, and that requires jumping through some hoops.
Why would you evolve that?
Way to put me on the spot.
Oh, sorry. I'm just thinking it's interesting all this diversity occurs because I think there's some protective elements to it, right?
I mean, okay, so if you're talking about this being a mechanism of evolutionary advantage or evolutionary development, you imagine that you would stress test these systems in an organism. And then basically, if the system isn't capable of functioning under really, really high stress, then those cells get deleted. The problem is that oftentimes you'll end up having a bunch of cells that can't deal with it, and that's when you get systemic disease. Because if you think about something like COVID, if you have just one or two cells that are infected in such a way that they can't actively respond to the viral load and their protein machinery is messed up, but all of your other cells are fine, it's highly unlikely that you're going to have a serious response. It's when you have a systemic problem that prevents your machinery from being functional in the face of stress that you start to have problems that are systemic in nature.
Well, yeah, and remember, the viruses didn't evolve to kill things. They're generally just trying to do their job, whatever that is, even though we don't understand it for the most part. They are definitely trying to do stuff, and they're only going to survive. I mean, it's very evolutionarily disadvantageous to kill your host, right? It's not a good idea.
Unless your host is literally every living thing on the planet, in which case
you
don't
can kill
have a
some of them, but you don't want to kill all of them, right?
It's true.
So even like these bats with Ebola aren't dropping dead. Like some, these things are there because they did their job the best, which is to make more of themselves, essentially.
Yeah. This is the graphic that I was talking about that basically talks about the 7 different classes of viruses. This is called the Baltimore classification, which unnecessary to know the name of that. But basically, the point here is that eventually all roads are going to lead to mRNA. mRNA is basically the key that is necessary to make proteins. So it doesn't matter what genetic material you start with, you're going to have to figure something out in order to basically get to the mRNA. Let's see. What's this guy?
This is an example of some ORFs. So I just maybe we can save this until we get to that section of the paper. But in the paper, that's annoying.
I got it.
All right. So in the paper, they mention ORFs, this word ORF, which is a open reading frame. Goodness, can you just mute that ringer? Yeah. So this is kind of interesting. So depending on where you start transcribing a gene or translating an RNA, as it turns out, these are the same sequences. You're just starting at different points in it. And so that's really interesting because you get completely different amino acids, and you can get different messages like stop, start, those sorts of things, depending on where you start reading the gene or the transcript. And so one of the things they mentioned in this paper, which we'll get into in a minute, is that there's 2 different open reading frames, and that there's actually I believe there's a more pathological version of it that's associated with sort of worse outcomes. And that's kind of interesting.
Mm-hmm.
I just wanted to familiarize y'all with that before we get into it. What?
It's fascinating.
On other websites. We can just click that. Maybe. All right, so this is kind of cool. So the first thing, we're going to tell the story. The goal of this paper is to tell the story of how how the virus works, how it gets into your cell, and how it replicates. If we can understand that today, then we'll be in a good position to understand how your body meets it going forward. And so this paper, I think, starts to touch on some treatment options and stuff, but we're not going to really worry about that too much. We really just want to focus on what is physically happening inside of the cells when the virus when your body meets the virus for the first time or second time or whatever. So this is the first thing that happens is the virus has to make entry into the cell. And let's talk about that a little bit. And the paper goes into it. We can maybe jump back and forth at this point. And so let's watch this little video. It's just a cool animation. Hopefully it'll stick it in your brain. You show a sound?
Contact. This is the moment when infection begins. How does infection happen? You see the tiny shapes there on the virus particle's surface? They seem like random blobs. In reality, they are tiny keys, proteins that have evolved to the perfect shape and size, enabling them to unlock these receptors on the surface of this lung cell. Like a smile and a handshake, the virus deceives this lung cell and is welcomed in, where it will command the cell to make more virus particles.
Indeed. All right, so let's look at that mechanism in a little more depth. All right, so this will give us a little bit more mechanistic details of what's happening.
SARS-CoV-2 is the virus responsible for COVID-19. The surface of the virus is studded with spike proteins, colored teal, membrane proteins, colored yellow, and envelope proteins, colored red. A cross-section shows the viral RNA genome in orange, and the nuclear capsid protein, which protects the RNA, in gray. An individual can be infected with SARS-CoV-2 after inhaling airborne viral particles. Upon entering the airways, the virus particles encounter respiratory epithelium cells in the trachea and lungs. The SARS-CoV-2 spike protein recognizes a membrane-bound protein on the surface of the cells known as ACE2, shown here in purple. Once bound to ACE2, the spike protein is unable to open, close, and bend freely, locking it in place. Another cell surface protein called transmembrane protease serine 2, shown here in orange, cuts the spike protein in a specific location. These segments of the spike protein fall away and expose previously hidden parts of the spike. The spike protein can then undergo a series of dramatic conformational changes. During the first stage, the spike protein inserts itself into the cell membrane. Once inserted, the spike protein folds back on itself, pulling the membranes of the cell and virus together so they can fuse. After fusion, the viral RNA is deposited into the host cell, where it hijacks the cell's machinery to produce more virus particles.
Love that one.
Also note that they discuss it as a threat.
Can we copy that link into the presentation for next year? All right, so this is interesting. So the ability for it to actually change shapes once it locks in and makes contact is mentioned in the paper as being part of what allows it to be so pathogenic. And they also talk about how this has to do with any virus's ability or inability to cross between different species boundaries has to do with unlocking this gate. And so this has actually been used. A similar mechanism has been used synthetically in laboratories to actually increase the pathogenicity of viruses. And so this is a very well-known mutation that might play into why this is such a nasty virus and that it can get into us across species.
You want to watch this one too?
Let's do the first section of the paper, and then let's watch this once we get to the replication stage.
Should we take a 5?
All right, so sure, if you want.
Yeah, let's take
All right, let's take a 5-minute break so that your brains will be ready to go.
Nimble. We come back, we start on the paper. See you at 38. All right, so video about the overview of SARS-CoV-2 replication, and then we go into the paper.
Sure.
Coronaviruses are a family of viruses that cause illness in humans and animals. 7 different types have been found in people, including those responsible for the SARS, MERS, and COVID-19 epidemics. Members of the coronavirus family have sharp bumps that protrude from the surface of their outer envelopes. These bumps are known as spike proteins. Spiked proteins are what give the viruses their name. Under the microscope, those spikes can appear like a fringe or crown. The spike proteins are located on viral envelope, which is the outermost layer found in many types of viruses. Other proteins are also inserted into the lipid membrane. Coronavirus's genetic material consists of RNA and not DNA, and is covered with only one major structural protein, often referred to as the nucleocapsid protein. For replication to occur, the virus has to first enter the host cell and bind to receptors found on its surface. Of these surface receptors, ACE2 is what coronavirus's family members, such as SARS-CoV-2, use to gain access. ACE2 is also expressed on the surfaces of other cell types such as those of the lungs, thereby explaining why this virus is responsible for respiratory system infections. One way the virus enters the host cell occurs when the spike proteins bind to surface receptors, and the virus is engulfed by the host cell. The spike protein is then cleaved, leading to the merger of the viral and host membranes, and entry of the viral RNA into the cell. A more direct way is when the virus fuses with the cell membrane and depositing its genetic material directly in the host cell cytoplasm. This genetic material contains all the elements required for making more copies of the virus. The code for making the spike protein is here, as well as the instructions for producing the nucleocapsid and other structural proteins. Most of the RNA, however, codes for the replication machinery itself, and it is this machinery that is the first element produced. To translate its RNA into viral proteins, the virus uses its host cell's ribosomes. The initially produced viral proteins are referred to as polyproteins because their size is huge. And they will need to be cut into several smaller proteins, including the 2 proteases, which will do the cutting itself. Many of the newly produced proteins are then inserted into the various membranes inside the cell. Here, we can see the viral RNA being translated in the endoplasmic reticulum. In essence, the virus has hijacked the cell's machinery and created a new machine which can now multiply its viral genome. This machine is referred to as the replicase transcriptase complex. Replicase transcriptase complex uses the viral RNA, also known as plus RNA, to create a complementary copy called minus RNA. This minus RNA is then copied to make more plus RNA, which can be packaged into new viruses. The next elements created are the viral structural proteins. Here, replicase transcriptase complex first makes small minus RNA strands. This is in contrast to ribosomes directly translating the RNA as seen earlier. Different small strands, including the 4 types shown here, are in fact made in this manner. The replicase transcriptase complex then makes copies of the small -RNA strands to produce small plus strands. These strands are now called subgenomic mRNA. And the host ribosomes can now translate these subgenomic mRNA into viral proteins. Among them are the nucleocapsid protein, the spike protein, and 2 additional viral proteins found in the viral envelope. Three of the structural proteins types shown here are made by ribosomes on the ER, where the proteins get incorporated into membranes, whereas the nucleocapsid protein is produced in the cytoplasm. This way the host cell is turned into a factory for making viral elements, the production of which occurs in networks of double membrane vesicles that form in these infected cells. While the nucleocapsid proteins code and protect the viral genome, the structural proteins move through the internal membranes of the cell, on their way towards the cell membrane. Their first stop is the ER-Golgi intermediate compartment, where viral genomic RNA and membrane-embedded viral proteins merge to form viral particles. The virus hijacks the host cell's secretory machinery, moving newly created viruses through Golgi and finally to the cell membrane. Vesicles full of viruses fuse with this cell membrane, releasing new coronaviruses outside the host cell. This replication cycle repeats itself as these new viruses encounter other cells.
Excellent.
All right, so there we have an overview, at least a pictorial overview of what the majority of this paper is going to be talking about. One thing that's really striking about this particular replication process is that the first thing the virus does is it makes this piece of machinery that it's going to use to build itself going forward, which is quite interesting. And the paper refers to this as it's a non-structural, let's see here.
A NISPs?
Yeah, I hadn't seen this word before. But this is from what I understand.
Non-structural proteins. Hold on, before we get into this, Wesley asked a really good question while we were watching the video, which was on the subject of spike proteins. And that is whether or not all viruses have spike proteins. And the answer is no. Coronaviruses are named coronaviruses because of the spike protein structures that are on them. Most other viruses will have some kind of receptor binding surface structure that is what allows them into the cell, but they will not look identical to the spike protein, and they will not behave identically to the spike protein. So spike proteins are particular to coronaviruses.
I guess HIV has them too.
The same ones?
They have something called a spike protein also, but it's different. It's not the same one.
I think that spike proteins largely are coronavirus specific, though. And then there are other, like HIV, if we go back to the chart that we were looking at in the presentation, it is a total outlier in the group that it's classified within. And so it might be that it has accumulated some kind of chimeric structure from infection with multiple viruses or something like that. But I think that spike is particular to the coronavirus.
It says its spike is like a amalgamation of a bunch of different protein complexes that are separate.
Oh, there you go.
That are encoded in its envelope gene.
All right, so back to SARS-CoV-2. So basically, what you have is that you begin with the viral particle. And see, they're very careful about this. Viral particle, not virus. So you have the viral particle that is carefully packaged. It's on the outside of the cell. It has the spike protein, the envelope, the membrane, the nucleocapsid, and inside all of that is the single-stranded RNA. It interacts with the surface of the cell. We watched how this works. And then it comes into the cell, at which point it is uncoated. and the first transcription event occurs, which is the fact that you get this ORF1A and ORF1B, which together make 15 to 16 proteins, 15 of which are involved in this replicative non-structural protein complex. So I don't know much about this in other viruses, but this non-structural protein replication complex I think is kind of unique.
I think so too. Last time I studied it, I'll have to get back to you on that, but I think it was one of the striking features of this virus is that it makes its own machinery immediately. A lot of them will continue to use the host's ribosome to produce all of their goods. That doesn't necessarily appear to be the case. These NISPs are allowing for the virus to duplicate its RNA, which will be extremely important in creating more of the viral particles down the road.
There's also this kind of other extraordinary aspect, which they basically create this entire complex that's part of the ER, which is this double membraned space where they're isolated from the rest of the cell, and it's able to go into overdrive, just producing more and more and more of the genetic material. This is a limiting reaction, right? So the minute that you decrease the amount of genetic information available, you're going to just start releasing empty viral particles because you just don't have anything to fill them with. And so you have to find a way around the sort of the protective mechanisms of the cell in order to actually be able to produce so many copies of this genetic information, which can then go on to produce all of these other structural genes that are super important for being able to actually assemble the thing. And so you secure yourself away from the surveillance system of the cell by putting yourself in this corner.
Actually, the next figure has to do with coronaviruses in general, which again underscores the fact that a lot of the coronaviruses are... So prior to this, prior to relatively modern times, like the MERS epidemic, the SARS, obviously the present pandemic, coronaviruses were thought of as the common cold, and they didn't actually lead to severe pathology in very many patients. I think that's sort of the take-home message from this figure about the history.
And you can see that the timeline is pretty... This is not mentioned is the non-linearity of this timeline, right? So the first coronavirus is discovered in 1931, and this is basically this avian virus. And then you have a gap of 35 years before you discover another virus. And then you have really not a lot going on for another 20 years, and then you start to see that these things tick up and become much more frequent. And there's two options about this. One is that your surveillance is increasing, right? So when you're looking at things that are happening in 1931, you're looking at something that cannot be viewed with the naked eye. It's pretty difficult to work with. Not a lot of labs are focusing on this. It might very well be that people are dying of coronaviruses, but there's so little surveillance and so little ability to surveil that there's no connection really between the chicken virus and the human virus, if there actually is a case where it jumps species. And then as you start to get closer to the present day, your surveillance techniques, your ability to manipulate these viruses becomes stronger. Your cost of doing sequencing falls, right? Because up until very, very recently, it cost $1 million to sequence a genome. Up until very, very recently, it could cost $1,000 to run a sequencing reaction on something that you have in a sample vial. Which means, and then if you actually wanted to take some kind of excerpt or some kind of extract from a person and be able to actually study, does this extract contain the virus? You have absolutely no ability to do that because that's going to cost you thousands of hours by many people to actually be able to isolate this. Like every time that you isolate the viral particle that is the causative agent of some disease, say Ebola, there's a team of people working on this full time, trying to go into the fields, collect the samples, go through the samples, isolate the thing, image it. So we've literally only in the last maybe 20 years have even gotten to the point where we're able to surveil this. And so it's very, very important to look at all these things that we find out about viruses and the way that they behave and the way that they affect us, and recognize that our understanding is biased by novelty. It is biased by the fact that we have just started looking, and there has never, literally never been a virus with so much attention paid to it as this one. And so whatever we're discovering now is not necessarily a generalizable factor of all viruses or of all viral particles, because they haven't largely been studied with enough detail to be able to carry this into generalities, is what I would say.
Yes. So.
All right, so here we're starting to talk about cleavage sites, and we're starting to talk about homology.
And diversity.
And homology and diversity. And so you have areas that don't have to maintain particularly strong homology, and you have areas that have to maintain pretty strong homology. And so looking at this part B here, which I have literally never seen a figure that starts with B and goes to A, but but bless them.
I didn't notice that.
I think that what they're trying to say is that A is the centerpiece of the figure, and then you, like, it's a zoom out to B, and then it's a zoom out to C. But still, it's crazy. So right here, we're looking at this cleavage site between the S1 and the S2 domain. So if you remember, when the viral particle is entering into the cell, what's happening is that there is a cleavage that occurs, which exposes the base of the spike protein. And when it exposes the base of the spike protein, that's when you start to get the spike that reaches down into the cell and causes the vesicle, the membrane of the viral particle and the membrane of the cell to fuse. And so you would expect that site to be relatively conserved, because if you have related viruses and they're all getting into the cells the exact same way, you would expect that that site is going to be fixed, because if you start to mess with that site, then you lose your ability to actually get into the cells that you're trying to get into. But as it turns out, when you start comparing SARS-CoV-2 versus all of these other all these other virus strains, and we'll come back to sort of the phylogenetic tree of these a little bit later, what you end up really seeing quickly is the fact that this cleavage site appears to be unique in this coronavirus. None of the other ones have the same site. Like, it's even reported in this very, very strange way, where it's saying that you have this 100% similarity here for all of these sites, and that's true. But then when you get here, you're obviously no longer at 100% similarity. Neither are you at this site. And so there's a couple places in this paper where the figures are somewhat misleading, just based off of how they've done their alignments, because what they've done here is they've opened gaps in order to create alignments that are missing in other places. And so, for example, here you have these two proline residues which exist in this RMYNO2 and the SARS-CoV-2, but none of the other strains have that same residue, despite the fact that this is all this cleavage site. And it's a little bit surprising that this has so much discontinuity. And I don't know if you were sort of focusing on some of the research that was coming out right at the beginning of the pandemic, when people are asking the question of, well, where did this virus come from if this is what its cleavage site looks like? Because the lack of homology here led a lot of people to be like, well, it looks like it could have been engineered, because to this day, we have yet to find a coronavirus that has the same cleavage site. And viral evolution works through chimeric recombination. You wouldn't expect to develop a totally novel site unless it has been somehow designed. It's possible that it's mutated within people once it's already gotten inside of people, and there's somebody that had some kind of permissive receptor that allowed it in, and then it mutated accidentally. And so And this is still very much unclear. Then you have this other domain, which is the ACE2 binding domain. And again, you see that there's some homology over the course of this. Again, I'm looking at this figure and it's like they're not pointing out where there's, I guess it's less than 60% homology. Never mind, I got it. And so you see some homology here. So it says SARS-CoV-2 ACE2 contact residues. And what this is saying is that as you look, oh, by the way, does everybody know what it is that we're looking at here? I'm just like basically nod your head if you understand what these letters represent. Okay. So for those of you who are not quite there, what we're looking at is we're not looking at the genetic code, we're looking at the amino acid code. So each and every single one of these letters is basically a stand-in for the amino acid that is at that position. Amino acid sequence and function is thought to be deeply related to the degree that you can do computational predictions about protein similarity based off of the presence of given motifs. This is a pretty standard research approach where you're like, okay, so this protein has this motif and this protein has this motif. It's entirely possible that they're related in function. And when you start messing with protein residues, you start messing with functionality because you start messing with the shape. And so there's some substitutions that you can make that are non-destructive, where the amino acids are kind of shaped in the exact same way as the amino acid substitutions are substituting for an amino acid that's shaped in the exact same way as the original amino acid. But here we're seeing some pretty significant substitutions at these contact residues where there doesn't appear to be a consensus for what kind of residues are necessary to actually come in contact with ACE. And that is, again, pretty strange because you would expect to see more homology from viruses that are so closely related to one another. And this is actually a very interesting question that if somebody wants to do a research project on it, would be to look into what are the natural mechanisms or unnatural mechanisms by which the sequence could have evolved to look like this. Because there's been a lot of research that's been done on this. I think that it could be an interesting project.
Like what would it be being selected for?
Or what was being selected for based off of amino acid substitutions, yeah. And that would be pretty, if somebody's really into biochemistry, that would be the the way to go.
Which part of the receptor it had more affinity for or something like that?
Mm-hmm.
Yeah, I think this also highlights just like the great deal of diversity that's kind of immediately apparent in these coronaviruses, which makes, which when we start talking about developing vaccines, becomes extremely problematic because you're talking not just about one object that you're trying to alert the immune system to, but an entire class of objects with very different shaped functional regions on their surface.
Mm-hmm.
So
box 2 over here is basically just a family tree of the different coronaviruses. And so the point of this box is basically to draw your attention to the fact that the coronavirus that is most closely related to the one that has been plaguing us since 2019 is a coronavirus that is a bat coronavirus, which does not seem to have sequence homology at the receptor binding domain. And that is what is causing some people to not understand the evolutionary past of this virus. And so as you look through all of these different strains of coronaviruses, you find that, as you go, it's not totally a linear progression, but it kind of is. As you start to go back farther in time, you start to be more distantly related. So you start with the 2009 virus. Up here you cluster viruses that were isolated 2019, 2013, 2017. So they kind of cluster relatively close to each other. Then you still have 2 more here that are 17 and 15. But then as you start to move away from this 2019 date and you start to move farther into the past, you're generally moving further and further away in the evolutionary tree. And that complicates your ability to say something about the provenance of the virus, because what your readout off of this is just the fact that the virus has evolved over time and that your sampling isn't particularly good to have been able to track the evolution of it over time. You can construct this sort of evolutionary tree for literally any 2 sequences that you have. You can go into a program, you type in your 2 sequences, and it will produce to you this evolutionary tree that you can put, it gives you the ability to put these quantifiable numbers on it to be able to say that, like, look, this is the sort of the evolutionary distance between these 2 things. And so you can take it with a strain of like a human protein and compare it to a jellyfish protein, and then compare it to a shark protein, compare it to like all of these different things, and it'll spit you out this computational map of how closely related these things are.
This is how people end up saying that mice are 99% the same as humans or something like this as well.
Yeah, because they're not looking at like every single letter in the mouse genome, right? They're like, okay, so we're going to take this protein, or we're going to take this gene for this protein, and we're going to compare it across species. And they're so closely related between humans and mice, but in reality, you're just looking at one protein, maybe a couple of proteins.
It also, these trees don't necessarily tell us, unless they make the assumption that the only way that these sequences could be homologous is if they had a similar ancestor, which is, a reasonable assumption, but it is an assumption that sort of ignores the possibility of convergence, which is where there's pressure in the environment that results in different organisms coming up with the same feature that are not really direct family of one another. So, for instance, it's a good example. Sharks and whales are a great example, right? They both have tails and flippers and swim around in the ocean. They're really not that closely related to one another. They both came up with tails and flippers. And so this kind of thing can play out in nature, and it can confuse some of these analyses as well. So this is by no means definitive, but it does give us some idea of, like Dr. Benbury said, the origin, to location, and at least the species from which this is most likely an amalgamation. In the discussion section of the paper, they go on about different fusion processes that are known to have occurred that could have resulted, sort of the shuffling of different coronaviruses together that could have resulted in this newer one. These are entirely speculative. We don't know for sure whether that's happened or it hasn't. So
down at the bottom of this figure, you also have another identity map, which is basically carving up the different constructs that are made off of this genome, and basically showing how similar they are versus how not similar they are. And so some of these, you can see that the nucleocapsid protein maintains pretty strong homology over the course of the entire phylogenetic tree. You can see that when you get to the spike protein, you're starting to get, especially the beginning, the first part of the spike protein, things are a little bit weird.
And this is what we're vaccinating against too. Is obviously this entity.
And so this is actually a really interesting point, because looking at this, what you see is that the S1 domain is highly, highly, highly variable. Unless things are, if things are the same between relatively distantly related viruses, then you would expect that there is some kind of pressure holding them to be the same. For example, the nucleocapsid protein has a very specific shape-related function where if you start messing with it too much, you begin to destabilize its ability to do its job. And so that makes a lot of sense. It is maintained really, really strongly across all of these different strains. If you look at the envelope protein, the same thing. If you look at the membrane protein, it's a little bit more variable. Like these faraway strains are getting a little bit more diverse. But then if you look at the spike protein, you start to see that there's strong variability in the spike protein. And that is what creates a lot of conflicts when you're looking at vaccine escape variants, or if you're looking for efficacy of vaccines over a long period of time, because how can you possibly have efficacy of a vaccine for something that you can see evolutionarily shifts relatively quickly?
And what makes matters worse is the S1 is actually the protein that's going to be encountering the environment. It's the most external surface of the virus. So when you're making antibodies against the virus, they're going to be against the things that are touching outside first. This is a crisis. This is a struggle for the technological development.
So basically, these phylogenetic analyses, whenever I see them in papers, I appreciate what they're trying to do, because you want to be able to reach out to something meaningful in order to be able to say that like you have this understanding of how things are related to one another. But when you look at this sort of this heat map of similarity, you begin to realize that there's clearly some kind of chimeric recombination that's happening in, for example, this like protein number 8 looks like it can be shuffled from different viruses. You can also see that depending on which part that you look at, you're actually looking, your similarity maps would be very, very different.
So this is a pretty hot topic. I just found a paper right away about some groups that are arguing for the exploitation of the S2 unit because it is more standard. I guess it's just harder to get an antibody in there, just if you can think about it.
Your timing for being able to get the antibody in there would be much smaller, right? Because you would only be able to get the antibody in there once it had already encountered a cell.
When it's opened up, you mean?
Yeah, exactly. But I imagine that that's a very crowded, very brief period of time. So you wouldn't necessarily be able to get in there. All right, so moving on from this, we have this figure, which is basically just showing you all of the different ways in which the RNA is processed in order to get the chunks that you want out of it. And so these, so it's called a polyprotein because you have all of these different reading frames that are shifted within the protein in order to get the things that you want. And so in some way, it's actually a shockingly effective information storage strategy, which is like if you think about your hard drive has a way of storing information that is in zeros and ones. And so you begin reading at the beginning of the file and you read all the way through and everything is delivered in zeros and ones. But if you could find a way to bin those zeros and ones into groups of 3 and then be able to tell your computer that like, okay, so this is your text file, or this is your file that contains all of the pieces of information. But if you start reading from position 1, you get one program. You start reading from position 10, you get another program. You start reading from position 25, you get a third program. You would be able to maximize storage and information processing to a massive degree. But this isn't how we do it because life does not, or human engineering usually doesn't imitate nature. But it's always really interesting to look into viruses and into the genomic organization of other beings and occurrences in order to be able to see novel ways of doing things. Polyprotein processing is almost like intron splicing. Like I said, that only mammals have introns, where you have this RNA strand that is processed in a differential way in order to be able to produce this thing that you want. And polyprotein processing is kind of like the viral version of intron management, where you're basically going through, and what it's showing here is that all of these arrows are basically feedback points, where, what does it say here? I just was looking at it.
By the way, I don't know if this is addressing your question from earlier, but we can do this in a second. But these 2 polyproteins are the results of a frame shift at the ribosome, whether you get the PP1 or PP1A or PP1AB. And I was thinking that had to do with the frequency of those was in some way was some way indicating the pathogenicity, but I can't find that now. I might have seen that in a different paper. Did you find the code there for the,
I have.
the red thing mean?
So these are basically proteolytic releases of proteins that are attaching here in order to actually produce to replicate the genomic material. And so basically there's these two proteins that are incorporated in this strand of genetic information, whose job is basically to manage and to make sure that things don't stick to the wrong place in the replication system. Like ribosome, and this is kind of insane because it is basically a parallel genomic processing factory to the ribosome to the DNA replication complex of the cell. And the way that it works is it basically stops the DNA replication of normal processes because they have all of these inhibitors that they produce, and then goes in and is basically like, we're going to take all of the resources and we're going to do it all ourselves, and we're not going to need to recruit any of the cell's proteins in order to be able to do it. And so it has all of the necessary pieces of it, right? So there's a couple of these chunks that people don't know what they're doing yet. But then that's going to be, I can't even imagine the difficulty of figuring that out.
That's a good research project.
Somebody who's done protein informatics.
You have the methods for how they figured out the other ones. But yeah.
So the method for how they figure out the other ones is they normally take the sequence and then they put it into something called either GenBank, or there's a protein database, and it spits out. Yeah, it spits out that they're like, other proteins with this sequence do this thing, and they're like, done.
That fits into our model, or it doesn't, right?
Exactly.
You have to have some idea of what needs to be done by the virus, and then sort of put it in that bin.
Mm-hmm. But so you have your own RNA-dependent RNA polymerase. You have this nucleotidal transferase, which means that you're perfectly capable of just assembling your own genome. You don't need anybody. These are just all of the genes that are necessary for basically turning off the rest of the cell, activating your own copying, and making sure that the vast numbers of you can get out of the cell. All right, so then we have genome replication from full-length negative-sense genomic RNA. Coronavirus replication and discontinuous transcription. This is a schematic depiction of coronaviral RNA synthesis. Full-length positive-sense genomic RNA is used as a template to produce both full-length negative-sense copies for genome replication and subgenomic negative-sense RNAs to produce the subgenomic mRNAs. So what this is basically saying is that you're using this first template, which is the positive sense genomic RNA, and you are producing other templates off of that are going to allow you to process other parts of either your replication complex or of the other parts of the virus.
It can also be fed right into the ribosome.
Exactly. And so what happens is that there's this leader sequence, and the leader sequence at the specific place is what allows for it to produce the necessary RNA that can be fed into the ribosome. And basically the real point here is the fact that you have this discontinuous transcription during the synthesis of the negative sense sgRNA, which is that instead of feeding you out this one piece of material, there's all of these attachment points where the machinery knows to attach in order to be able to give you the necessary transcript that's going to give you the form of the protein that you want for being able to assemble all of the pieces that you need for the virion to be able to leave the cell.
Including more positive sense RNA.
Mm-hmm.